Hello students, welcome to the class of Biochemistry. Myself Dr.R.Surekha, lecturer, Department of Biochemistry, SRM Dental College, Ramapuram. Today we are going to discuss in detail about copper. The minerals are classified into macro mineral and micro mineral based on the daily requirement. If the requirement of the mineral is greater than 100 mg per day, it is referred to as macro mineral. Example, calcium, sodium, potassium, magnesium, phosphorus. Whereas the requirement of the mineral is less than 100 mg per day, it is referred to as micro mineral or trace elements. Example, zinc, copper, iron, iodine, fluoride, etc. In this lecture, we're going to discuss in detail about the sources, RDA, functions and deficiency disorders of copper. As we discussed earlier, the copper is an essential trace element and it is involved in the proper functioning of various organs and metabolic processes. The total body content of copper was found to be 100 mg and the copper was found to be widely distributed in all the organs. Especially it is found rich in liver, kidney, cardiac, muscles and bone. Let us discuss the sources of copper. So this slide shows the wide distribution of copper in plant and animal sources. So the liver, kidney, meat, egg yolk containing a highest concentration of copper. Also the cereals, green vegetables found to have a copper concentration. The sources, especially the invertebrates, are the richest source of copper. Example, octopus, crab, prawns, shellfish. So these are all the various invertebrates which has highest concentration of copper in it. Milk is a poor source of copper as shown in the slide. Let us now discuss about the RDA of copper. The adult is advised to consume 2 to 3 mg per day of copper. The children 0.5 to 2 mg per day of copper. Since the requirement is less than 100 mg per day, the copper is considered as a micronutrient or a trace element. Coming to the absorption and the excretion of copper, like any other micro element, the copper is absorbed in the duodenum of small intestine. Once the copper is absorbed, it will be transported to the liver via blood. To the blood, the copper binds to the plasma albumin and transported to the liver. In the liver, the copper will be either stored or will be transported to the peripheral tissues. So the storage form of copper is ceruloplasmin. So 90% of copper is available in the body as a stored form, that is as a ceruloplasmin form. And 10% will be found in the transport albumin. It just binds to the albumin and gets transported to the various peripheral tissues. Excess copper is excreted mainly in the bile. The normal plasma concentration of copper is 100 to 200 mg per dl. Out of 100 to 200 mg, 90% of copper is delivered to the liver where it gets stored as ceruloplasmin. 10% is available in the blood bound to albumin. The total concentration of serum ceruloplasmin is 25 to 50 mg per dl. There are various factors which promotes copper absorption and decreases copper absorption. The factors which influences copper absorption is metallothionein, which is a metal binding protein which increases the absorption of copper. Here is the factors which decreases the copper absorptions are phytate, zinc, molybdenum. So these components, they just compete with the copper absorption and decreases its absorption. Let us now move into the functions of copper. Copper acts as a cofactor for several enzymes. Hence, the copper is mainly involved in the metabolic reactions. So these are all the various enzymes require copper as a cofactor. Delta amino levulinic acid synthase, ascorbic acid oxidase, monoamine oxidase, superoxide dismutase, tyrosinase, catalase, cytochrome oxidase, phenol oxidase and uricase. So these are all the various enzymes where copper acts as a cofactor. As we seen in the earlier slide, The copper acts as a cofactor for ALA synthase, that is delta aminolevulinic acid synthase, which is an enzyme involved in the heme synthesis. So the copper is very essential for the synthesis of hemoglobin because the ALA synthase requires copper as a cofactor. Ceruloplasmin, which is a storage form of copper involved in the ion transport. Because the ceruloplasmin exhibit a copper dependent oxidase activity, otherwise called as ferrooxidase activity. The function of ferrooxidase is to oxidize the ferrous form of ion, that is Fe2 plus, into ferric form of ion, that is Fe3 plus. So the conversion enabled the binding of a protein called transferrin to the ferric ion, thereby enable the ion transport. Thus the ceruloplasmin involved in the ion transport assistance just by converting the ferrous ion into ferric ion. Then the ferric ion can bind to transferrin protein and can transport it to the organs. The collagen and the elastin cross-linking is also enabled with the help of copper. The connective tissue protein called elastin and collagen has the highest concentration of lysine residues. This lysine residues will be converted to allyl lysine with the help of enzyme called lysyl oxidase, which is a copper containing enzyme. The copper acts as a cofactor for this enzyme. This conversion of lysine to allyl lysine by this enzyme leads to a cross-linking between the elastin and the collagen. These are the various other functions of copper. The copper acts as a cofactor for the enzyme tyrosinase to synthesize melanin pigment. The copper is also involved in the development of bone and the myelin sheath of the nervous system. Copper is also involved in the synthesis of a complex protein called hemocyanin in the invertebrates. This hemocyanin functions just like the hemoglobin of our body to transport oxygen. So that the presence of hemocyanin in the invertebrates makes their blood blue, appearing blue. Whereas in the human being, the hemoglobin appearance makes our blood red. Likewise, the hemocyanin makes the invertebrates blood as blue. So that is why the invertebrates have the richest concentration of copper, as we seen in the earlier slide, that is in the sources of copper slide. We have seen invertebrates has the highest concentration of copper because of the presence of hemocyanin in their blood. Deficiency of copper is maybe due to the three major causes, that is malnutrition. When the person has happened to consume a low copper diet, may develop a copper deficiency symptoms. Also the malabsorption nephrotic syndrome, that means the increased loss of copper in the urine or feces leads to a development of a deficiency disorders of copper. This slide shows the general deficiency manifestation of copper. Neutropenia in decreased number of neutrophils, hypochromic anemia in the early stages where the copper deficiency leads to a improper functioning of the ALA synthase leading to a reduced heme synthesis. Osteoporosis and various bone and joint abnormalities just due to the impairment in the copper dependent cross-linking of bone collagen in the connective tissues. Decreased pigmentation of the skin due to a reduced activity of tyrosinase, and neurological abnormalities due to the reduced activity of cytochrome oxidase. Those are the copper dependent enzymes. So when there is a low copper concentration, but that affects the enzyme activity, resulting in the development of deficiency manifestations. There are two inborn errors of copper metabolism. First is Menkes syndrome or kinky hair disease. The other one is Wilson's disease. So let us discuss detail about these two diseases. Menkes disease or kinky hair disease, which is a rare fatal X-linked recessive disorder, which is caused due to the mutation in the gene that encodes copper binding P-tape ATPase in the intestinal mucosal cell. That leads to a defect in the absorption and the transport of copper from the intestinal mucosal cell to the blood, resulting in the decreased intestinal absorption of copper. So this slide shows the clinical manifestation of Menkes disease. So this is due to the defect in the copper dependent P-type ATPase present in the small intestine. So the dietary copper is not absorbed or transported in the small intestine, resulting in the increased excretion of the copper in the kidney. So the reduced absorption results in the low copper concentration in the brain, skin and hair, bones, resulting in the development of Menkes disease. This two picture shows the Menkes disease or kinky hair disease. So this is a kinky hair seen in the child. So this is due to the malabsorption or the defect in the P-type ATPase in the small intestine, leads to a reduced copper absorption. The treatment of the Menkes disease helps only when it is started early. The treatment for this is the injection of the copper into the vein or under the skin. So here the treatment of oral administration of copper is not advised because the dietary copper or orally administered copper is not absorbed from the intestine. So it is very advised to inject the copper into vein or under the skin. Wilson's disease is otherwise called as hepatolenticular degeneration, which is a rare genetic disorder caused due to a mutation in the gene that encodes copper binding P-type ATPase in the liver, resulting in the poor transport of copper and secretion of ceruloplasmin from the liver. Due to the defect in the copper dependent P-type ATPase in the liver, the copper gets accumulated in the liver and subsequently in the other organs. This disease is fatal and death occurs at early age. So these are the various symptoms of Wilson's disease: the development of CNS disorders, hepatomegaly, arthritis, case of Fisher's ring, cardiomegaly, renal tubular dysfunction. This slide shows the clinical manifestation of Wilson's disease. Due to the defect in the P-type ATPase present in the liver, the copper gets accumulated in the major organs like liver, kidney, lenticular nucleus of the brain, eyes, leading to the development of copper toxicosis. This causes neurological symptoms, liver damage leading to cirrhosis, renal tubular damage, Fisher ring. So this is the Fisher ring, which is a brown pigment around the iris or the edges of the cornea. This is due to the copper deposition in the cornea. This is one of the main clinical manifestations of Wilson's disease. The copper deposition in the kidney results in the development of renal damage, along with increased excretion of amino acids, glucose peptides, hemoglobin in the urine. Thus, the Wilson disease causes reduced copper and ceruloplasmin in the plasma with increased excretion of the copper in the urine. Also, the copper gets accumulated in the major organs like liver, lens, brain, kidney, resulting in the development of hepatomegaly, Fisher ring, liver cirrhosis, renal damage, and mental retardation. The diet low in copper is advised to the Wilson's disease. Also, the administration of copper chelator is prescribed. That is, D-penicillamine is given to the patient, which chelates copper absorption or deposition. Thank you.